Thanks for coming out tonight. Nice, it's nice and quiet now. Wow, got a packed house, so super excited to see so many folks. Just want to kick off the meeting by just thanking the sponsors that make all of this possible: pizza, the beer. So thank you. If you are a pharma company or a large biotech and you have a budget, you'd like your name up here with these spectacular organizations, please contact us. This is really only possible because of the support of these organizations. So please help keep this community going and pitch in. Okay, so we've got Gabe Rocklin. Gabe, I think, probably needs very little introduction in this room. It's a packed house. Clearly, you all know Gabe is a rock star. He started in the Baker Lab as a postdoc, and I think the thing that I'm been most consistently impressed about with Gabe is trying to take a lot of the previous stage of protein design, which was really an art, and turn it into a science. And we didn't have enough data to really do that. And so I think Gabe has consistently pushed the boundaries of data set generation, making more measurements in high quality to allow us to really understand general principles for how proteins fold and how we can control that rationally. And I think every time I see Gabe talk, the experiments get bigger, the data sets get better quality, and we can learn more from them. So I'm super excited. Gabe, please take it away.
Thank you, Chris, for the introduction, the group for the invitation, and all of you for coming out. It's like awesome to be here. I was looking at all the speakers that you've had in this series, and this is like the most stacked seminar series in protein design. So I'm going to talk about. Good. I'm going to talk about large-scale study and prediction of protein folding stability, and also aggregation, which is a topic that I've never worked on before, and I think this is like the second time, or really the first time, to a live audience that I've talked about this work at all. So I'm excited to share it with you. So the first thing that I want to say, just building on what Chris said, is what really motivates me is the opportunities for applying multiplexed technologies to biophysics and improving modeling. So multiplex technologies, like technologies where we can measure large numbers of different proteins all in one pooled experiment. And so why is this getting so much more powerful? It's getting more powerful because all these technologies are getting cheaper, like the DNA synthesis to make all the proteins we want, DNA sequencing to analyze them, and proteomics to analyze them. And I'll be talking about two different projects, one that's based on DNA sequencing and one that's based on proteomics. And I was actually just giving this talk to like a very biophysics-y audience. And what I try to get across is that the idea of making a really large number of measurements where you maybe sacrifice a little bit of quality kind of cuts against some of the instincts of most biophysicists, because the field of biophysics is very often like, let's try to measure in the highest resolution, the greatest amount of detail that we possibly could. And so when we do these multiplex measurements, we do sacrifice a little bit of detail. But what we gain back is the ability to measure a vast number of sequences and learn how sequence influences all these properties. And then another, for a machine learning audience, what I want to emphasize is that what we can learn in modeling from biophysical measurements is actually really different from what we just learned from existing large data sets that are used in machine learning all the time, like databases of protein structures and protein sequences for evolutionary information. We learn really different things when we measure biophysical properties compared to just what we can already learn from these existing big data sets. One of the hypotheses motivating what I'm trying to do is that if we can learn principles of biophysics from these really large-scale measurements, that the deep learning models built on those data will be more transferable to other kind of low data problems. Like, I'm going to show examples of measuring folding stability for 2 million different proteins because we can measure folding stability really efficiently. But hopefully, models trained to predict folding stability will be more transferable to other types of problems where there's going to be much less data, like maybe energies of different conformations of a protein or something like that. And then finally, everything that I'm going to show you in this talk is about really small protein domains. So protein domains that are about 80 amino acids or smaller. Actually, there's one slide where it's not that. You know, again, I really like working with small domains. They're easier to scale. I think you can learn fundamental principles from studying small domains, and they're very powerful tools for improving computational modeling. But, you know, 3 years from now, I really want to be doing these scale of assays on all the kinds of proteins in the whole universe. And I think these small domains are great systems to prototype and to develop these experimental tools that we can then scale to all types of proteins. Okay, so I'm going to talk about two different things. I'm going to talk about large-scale discovery and prediction of protein folding stability, and then discovery and prediction of protein aggregation propensity. So for this audience, I think most people think about folding stability the same way that I do, the ratio of molecules that are unfolded to folded. And, you know, this is a really important property. It influences basically everything else about proteins, how they function, their aggregation, immunogenicity, you know, disease expression level. All these things are linked in one way or another to folding stability. And if you try to do machine learning on just one of these properties without kind of building in folding stability somewhere in your model's probably trying to learn some version of folding stability and figure out how that interacts with all these different properties. High protein folding stability is typically a key goal in protein design. It may look like this is already solved because often, you know, most of the AI designed proteins are stable to one extent or another. But I think this is always going to be a challenging problem, especially as we try to make more and more tailored active sites, because usually the functional elements in proteins are destabilizing to one extent or another. And the more complexity we want in these destabilizing elements, the better a model we need to have of folding stability in order to make it possible. I'll show you later on that folding stability typically does not correlate with the confidence of a predicted structure from a model like AlphaFold. AlphaFold and other tools will predict very confident structures for proteins that are completely unstable because they're answering a different question. They're answering the question of what's the most likely folded conformation of this protein, and not like how stable is that folded conformation compared to other things. And then finally, I hope that by measuring folding stability and creating good models of it, we'll be able to extend those models to other applications where data is more sparse, like energetics of binding or of populations of different conformations of a protein. Okay, traditionally, folding stability has been really difficult to measure. People always did this on purified proteins. And if you looked at all the literature up until a few years ago, there were maybe like 5,000 folding stability measurements in all of the published literature, depending on what you include. But we wanted to create a way to measure this at a really large scale. And so this was Kotaro Tsubeyama's work, who was in my lab for 3 years and is now a PI in Tokyo. I'm going to quickly review Kotaro's published work on this, and then I'm going to show you what we've been doing in the last few years, which is actually in collaboration with Galen Cho, who's standing here, whose dog everyone's been like awing over. Okay, so quick review of the published stuff from Kotaro, and then I'll show you some of the new stuff. So we developed a technique called cDNA display proteolysis, where the folding stability of all the domains are all measured in one mixture. So we start with a pool of DNA, and we use DNA oligo pools synthesized by Twist. All the domains that we've encoded so far are shorter than 80 amino acids, which allows us to fit each domain on one oligo. And we've actually worked with pools up to 900,000 sequences all in the same experiment. So we take this oligo pool, we express it, or we transcribe it and translate it using in vitro cell-free protein synthesis. And we use a technique that we did not invent called cDNA display, so that each protein is covalently attached to the cDNA that encodes that protein. This happens at the end of translation while everything is still on the ribosome. We reverse transcribe the mRNA back into cDNA and then covalently attach it to the protein. This is at the C terminus, and the N terminus of all the domains have an epitope tag that can be pulled down by antibody. Okay, this is all in one tube. We incubate this protein mixture with proteases. We actually do one set of experiments with trypsin and a different set of experiments with chymotrypsin. And what we found empirically is that these small domains basically only get cleaved when they completely unfold. That's not totally true. There are some domains that can get cut even when they're folded. But mostly for the small domains that we've looked at, they only get cleaved when they're completely unfolded. And that means that the cleavage rate basically tells you the global folding stability of these domains. So we pull at the end, we pull the domains down by the tag on the N-terminus, and then we sequence all the C-terminal DNA in order to learn which proteins survive this proteolysis treatment. And we actually do this at many, many protease concentrations, very low amounts to very high amounts in order to quantitatively resolve the folding stabilities of all these proteins. So this works pretty well. These are all the point mutants of protein GB1, all the single mutants. It's a 56 amino acid domain. And these are the folding stabilities measured with our technique. And these are the folding stabilities from the literature that were actually all measured in the same study. Alex Nisthal in Steve Mayo's lab used a robotics system to purify small amounts of each one of these mutants individually, and then did a chemical denaturation experiment. And you can see we get really good consistency between measuring everything at once by DNA sequencing and purifying each different protein and doing a traditional assay. Some of these points disagree because the fits from this study were really not good for proteins that didn't express very well. Some of these are probably errors in our experiment. And the ones down here are ones that basically couldn't express in sufficient quantities in their experiment. So this works pretty well. And we didn't just measure these mutants by themselves. This was actually done in a library of 700,000 sequences. So this is like the data quality that we could get even on this really, really large scale. So these are all the mutants of protein G. We also took all the mutants of small domains that we could find in the literature and compared them to our measurements. So across all these different small domains, some of which are all alpha, some of which are all beta, we saw really good correlations between literature values of someone purifying every single one of these proteins and the folding stability that we get from DNA sequencing by measuring all of these together. All of these individual fits actually have different y-intercepts because the literature measurements all come from different experimental conditions. Some people measure them at 45C, some people measure them at 4C, some people measure them, at different pHs and stuff. So we don't expect all of these to be quantitatively on the same scale. This is actually a good reason why it helps when you do all the experiments together, because then you're measuring everything under identical experimental conditions, which I think is really ideal for building machine learning models.
By the way, if you tend to interrupt.
Yeah, go ahead.
So that's okay. So for delta G from proteolysis, are you assuming? So I guess you're assuming a two-state model, but completely cooperative.
Yes. Yes.
And you fit that somehow to the rates of proteolysis, or
we compare the rate of proteolysis to the rate of proteolysis for a model unfolded substrate with the same number of cut sites as we built a model of proteolysis in the unfolded state based on tens of thousands of measurements. And then we compare our measured for all these folded domains to the model for the unfolded state. Sure, why not? Oh, hi, good to see you.
Briefly comment on the that's used.
Yeah, so what we did in this study was we did deep mutational scanning on about 450 different domains. So we took all the small domains that we could find in the PDB and a few hundred small de novo design domains that actually go back to my own postdoc work. And we did deep mutational scanning on all of them. And we published this set of 750,000 mutants a couple years ago. And so all these data are available. You can download them on Zenodo. And I've been really happy with the number of people that have downloaded them and cited them and so on. But this is really great if you want to understand effects of protein substitutions. But we had kind of the larger goal of, can we just take a protein sequence of a small domain and predict what that folding stability is? And just predicting the folding stability of a new small protein, there's never really been attempts to do that in the literature because you've got these really different energetic contributions, like you've got all the loss of conformational entropy of the protein folding up, and all the hydrophobics, and all the hydrogen bonding. It's a much more difficult problem than just how does changing this one amino acid change the folding stability, which is basically what people have done up until now. I'll talk about that in a second, but I just wanted to point out two of the exciting uses of the mutant data set that I was just talking about. Brian Coleman's group trained a model called thermo-MPNN to predict the effects of substitutions using all that data that I just showed you. And I think this is a really useful model. We use it in my lab for predicting stabilizing mutants. And then Frank Noe at Microsoft, whose model called BioEmu was just on the cover of Science, basically they wanted to create a generator of Boltzmann ensembles of proteins. And if you're going to create a Boltzmann ensemble of proteins, you need both folded conformations and unfolded conformations in the appropriate populations. And so in order to create this model, they trained it to predict folding stability on all the mutant data that I was just showing. So all these, I mean, we figured that people would start doing mutant predictors based on our data, but there's also other applications of our data that we didn't anticipate. And it's been really nice to see how it's been used. Okay, so like I said, mutant data is great for predicting the effects of substitutions, but really we'd like to just predict the overall stability of new proteins. And so this has been the collaboration that I just said with Yelin and Sergey, who were both here until Sergey had to run, I think. But anyway, this has been a total pleasure collaborating with Yelin. So thank you. Okay, so what did we do? Going back to Kotaro for a second, we put together a set of 2 million different small domains that are actually taken from the Magnify database, where we use the AlphaFold predicted models of all these different metagenomic proteins. Magnify is a database of metagenomic sequences, and we use those AlphaFold models to parse them into domains and then snipped out all the domains that were smaller than 80 amino acids. And actually, this data set was originally put together through a collaboration with DeepMind, so I should credit them. But then that collaboration stopped and basically at the point of generating this data set. So we took all these sequences of small domains, and then Kotaro measured folding stability for them using cDNA proteolysis. And so we ended up with this set of 2 million measurements. And if you cluster these by sequence, you get about 300,000 clusters. So it's a really diverse data set. And going back to what I said at the very beginning, if you just look at simple metrics, those simple metrics do not correlate with the folding stabilities that we measured for these domains. So here are some of the simple metrics that we looked at. The number of hydrophobic amino acids doesn't really correlate with the stabilities that we measured. The radius of gyration of the predicted models has a weak correlation with the folding stabilities. If you look at like structure prediction or sequence likelihoods, these don't really correlate with the folding stabilities either. So AlphaFold PLDDT or ESM-fold PLDDT are not higher than 0.35. Kind of sequence likelihood from ESM-IF or protein MPNN get to correlations of about 0.4. But if you take all these data and fine-tune some protein language models on them, then you can actually get really nice correlation. So up to 0.9 of Yelin's best fine-tuned SAPROT model. So using a data set of many diverse small domains, you can train a language model to accurately predict the stability of new sequences that are different from this, that are of the same general type, but are not related by sequence to the sequences that we're trained on. Please.
I'm wondering if for the AlphaFold 2 PAE and PLDDT,
yes,
for the zero shot stuff,
yes,
were those taken from the full proteins where before you parsed the domains, or did you refold the domains?
That's a good question. I'm not totally sure.
You refold the domains.
Refolded the domain.
Okay, that makes sense.
But also, like, I would not expect a strong correlation because these are going to be really influenced by the MSA quality and stuff like that, and they're not influenced by, like, does this have the right biophysical elements in order to fold?
Yeah.
Please.
Have you tried using thermo-MPNN to predict stability?
Thermo-MPNN does not actually have the ability to predict the stability of a sequence. It only predicts the effects of substitutions. That's the way that thermo-MPNN was created, to predict effects of substitutions. Please.
How that fix the actual value is close to the range.
So this is the SAPROT, the fine-tuned SAPROT model has an MSE of about 0.8 kcal. I don't know the MSE of all the other ones. The dynamic range of the measurement is not super wide, so we're measuring from about -1 up to 4 or 5 kcal. Well, here's the experimental range. So we measured some things above 5 kcal. Basically, if you just predict the median, you're going to get an MSE of maybe 2.5 or something, or 2 kcal. Oh, hi. Yeah.
You know, the old ancient algorithm, when it from a primary sequence gives you, stability measure.
Oh, no, we didn't try it, but
Yeah,
okay.
just curious how, I mean, I don't expect you to do better,
Yeah.
but given the range you have here.
Yeah. Okay, so I'm going to keep going, but I'm happy to keep discussing this after. I'll just say that over the really long term, I don't necessarily think that a language model is the best way to do a problem like this. I'm actually kind of excited about the SLAE model that Po-Soo Huang's group just put out. I think that would be cool to try. Anyway, but yes, but even with a language model that does not necessarily have a detailed 3D understanding of all the interactions, we can still, by training on enough data, predict folding stabilities of new domains pretty well. Okay, so we have this model that works on these domains up to 80 amino acids. Can we generalize this and predict folding stability for any protein? And the answer is basically no. But I still think this model is useful in a lot of contexts, and I'm going to show you a few of those right now. One of the first contexts is we tried using Yelin's model to predict or to see how it correlated with melting temperatures of nanobodies from this nanobody thermo database. And we actually got a reasonable correlation, even though, first of all, the thing that we're comparing to is like an aggregation temperature, which is not really a thermodynamic measurement. It's not a folding stability. And also all the nanobodies that we're evaluating this on are about like 100, they're 50% larger than any of the domains in our training set. So this is applying the model to something that's pretty outside the training set, and it's a completely different measurement. Like our model was never trained on temperatures of anything, but it has a reasonable correlation with the melting temperatures of these nanobodies. And, more importantly, I think that if you had nanobody specific data, this would be a really good pre-trained model to then apply to your new, more case-specific data. We didn't try that application yet. Another thing we did was we just took all the design mini binders out of the RF diffusion paper and asked if we rank these mini binders, these candidate mini binders by predicted folding stability, do we provide any enrichment of good binders versus bad binders from the original RF diffusion paper? And so these are the targets that they designed binders to in the RF diffusion paper. And for several of these targets, if you rank the designs by predicted folding stability, you actually get some pretty good enrichments of successful binders versus failed binders. So again, like we wouldn't expect perfect enrichment from this model because, of course, they're testing for binding, which is going to depend on how well these proteins fold and how well they bind their targets. But just applying a better filtering on folding stability, even with AI designed AlphaFold filtered protein designs, still gives you enrichment of successful binders versus unsuccessful. Okay, so this is cool new work that, so anyway, what that finish of this project is that hopefully this will all be pre-printed and all the data will be released in the next couple months. We have a manuscript that we're really excited to get out as soon as we can. But going beyond that manuscript, what we're now doing in my lab is trying to apply proteolysis screening to just any size domain and trying to increase the dynamic range of folding stabilities that we could measure. So one of the limitations of the technique that we've been using, where we use a gradient of protease concentrations, is that we can't really resolve folding stabilities higher than about 5 kcal per mole. So we're completely changing how we do these experiments to now use a gradient of denaturant, which is kind of similar to how you would do a traditional folding stability assay. Like you would have, a small amount of urea, a higher amount of urea, and so on. And so when we started doing these denaturant-based experiments. This is really preliminary. This has a long way to go, but we now see pretty good correlations with folding stability across mutants of these different proteins, and all of these proteins are larger than what we've ever looked at, and some of their folding stabilities are quite high. So the mutants of thyroidoxin, their literature folding stabilities are between like 7 and 10 kcal per mole, and we're not quite resolving. Well, we're resolving them somewhat, but we are measuring them to be really high stability, and likewise we can still resolve the mutants of things that are lower stability as well. So in the next year, I think we'll translate this assay to be able to measure folding stability across a really wide range, even for proteins that are much larger than what we've measured in the past. And this is Ted Littberg and Adun Obisesin in my lab. Okay, now I'm going to switch to a completely different story, which I've never presented before, so I'm excited to share this with you. I'm just going to poll the audience. Who in the audience has worked with a protein that's a pain in the butt and aggregates a lot? All right, pretty good. So protein aggregation is a big pain. I am not an expert in aggregation compared to all of you in the audience, so you have to forgive me. I'm interested to learn what things cause problems for all of you, because I think this is an area where multiplex assays could have some value. But I'll just quickly talk about two proteins. This is Neupogen. It aggregates at physiological pH, so it's formulated under a low pH. You really don't want to shake it that much, because if you shake it, then it'll start to precipitate, and that's pretty common for a lot of proteins. This is Avastin. It had to be formulated at a really low concentration, because at a higher concentration it starts to aggregate. You don't want to store it for more than 8 hours. So anyway, all of you know probably much better than I do that proteins are a big pain often, especially at high concentrations, and we have to do all these things to prevent them from aggregating. But it would be great if we could design sequences that were more aggregation resistant. And I'm not a formulations person. I don't know anything about excipients and so on, but I think we can still learn a lot about designing better sequences. There's lots of aggregation predictors out there in the literature. I'm sure lots of you have tried to use some of them. They seem like they're not super useful. Some of the examples that we found. Oh, so actually, before I go to that, why is aggregation difficult to study? So it depends on the protein sequence. It depends on the environment that protein is in, like the pH and other molecules around. It depends on stresses applied to that protein, like shaking or hot and cold and so on. All these different things interacting with each other. Of course, proteins can aggregate in multiple ways. They can aggregate from the folded state, from the unfolded state. They can form amyloid, or they can form more like folded aggregates. Aggregation is influenced by all these different conformational processes in proteins. Most studies of aggregation use purified proteins for understandable reasons, but that's a real roadblock to collecting a lot of data. Of course, proteins themselves are really complex. That's one of the things that's fun about what we do. And there's also pretty limited public domain, large-scale data on protein aggregation. I know lots of companies have surely measured formulation for many different candidates, but there's not a lot in the public domain except studies of cellular proteomes, like thermal proteome profiling, which is not exactly the best source of data for doing machine learning. We could talk about it if you're interested. Okay, so it's difficult to study for all these different reasons. And like I was trying to say earlier, current machine learning tools are not super successful at predicting this. Some of the examples that we found in the literature, this is a study that we found where people used computational aggregation predictors to try to design point mutants that would stabilize GCSF, which is the molecule of Neupogen. And they tested some of these computational predictors, and all the mutations that they designed actually made their protein more aggregation prone. And this is not shocking to people. What's actually shocking is to see this in the literature, because you often don't see, negative results. So it's kind of nice when people publish their failures as well. This is an example of understanding a what's the protein here? Beta 2 macroglobulin. that there's an inherited variant that causes this protein to form amyloid for people with that variant. And the Radford group wanted to understand if computational predictors could predict this variant or could predict other protective variants. So they actually tested about 70 different variants of beta-2 macroglobulin and found some kind of weak correlations with computational predictors. But it was very difficult for them to rationalize the experimental data that they collected. Okay, so our strategy was to create customized mixtures of protein domains and measure aggregation for all these domains mixed together using proteomics. And that's where the scale comes in. And all of this was done by my really amazing grad student, Sydney Martell, who did all the experiments for this project and all the computational analysis for this project. And she's going to graduate in the next few months and is looking for jobs. So you should all hire her. And she also funded her entire PhD through a really competitive T32 program and a fellowship from the Pharma Foundation. So everyone who's a member of the Pharma, thank you to them for supporting this work. Okay, so what did we do? We encoded diverse small domains in DNA oligos, just like what we do for the proteolysis experiments. But then instead of making those proteins through cell-free protein synthesis, we actually took the mixture of all the DNA oligos, clone them into plasmids, transform them into E. coli, and then instead of taking one colony, we scraped the entire plate of E. coli into a liquid culture, grew it all up together, and then purified all these proteins using a his tag, and then cleaved off the tag. So it's just like purifying one protein, except instead of purifying one protein, we're taking all these transformants in one culture, growing it all together. And we end up with a soluble mixture of all the proteins that we encoded in our original DNA oligo pool. And the proteins that we studied here were actually about 20,000 examples from the metagenomic library that I told you about before, about all the domains that we used for the folding stability studies. So it's the same type of diverse 60 to 80 amino acid domains, except now we're not tagging them with DNA. There's no DNA tag. We just have a soluble mixture of all these different domains. Please.
They 10,000 domains per library for the proteins that were eligible.
So we did these experiments on 2 different libraries that each were designed to have 10,000 proteins, and we included about 1,000 overlapping proteins so we could study how the library context affected those 1,000 overlapping proteins. Okay, so then we take this mixture of proteins and we exposed it to a few different stresses. So we heated it up to 50 degrees and 75 degrees and then cooled it down really quickly. And we also took it from pH 7 and moved it into a new buffer at pH 4. And then we took these different samples and removed the insoluble aggregates. So we actually did not do anything to look at soluble aggregates. We just removed proteins that became insoluble after these different stresses. And then we used TMT labeled proteomics to quantify how much of the abundance of all these proteins changed between our starting sample and the sample that we recovered from the soluble fraction after these different stresses. Okay. Okay, so this is the fold change of how much of these proteins were left after we did the stress. Some of the proteins seem like they increased just because of the normalization of how this works. But we don't gain any protein from doing this. But a bunch of proteins went down by a lot in the stressed sample compared to the starting sample. The 75C stress caused the greatest amount of aggregation, which is why we see the largest number of proteins with very negative fold changes. So if you have a fold change of -2, that means there's like 25% of that protein in the stressed sample compared to the starting sample. And a fold change of -4 means there's like 1/16 as much of that protein left in the stressed sample. So the 75C had the most aggregation, the 50C had a little bit less aggregation, and the pH 4 had the least aggregation. But okay, so we saw some aggregation. Now, like, what did we learn? So going back to the question that was just asked, do proteins aggregate differently depending on which mixture they're in. And one of the cool results that we got was for the different stresses that we looked at, we saw basically the same level of aggregation for the 1,000 proteins that were in one library of 10,000 versus the other library of 10,000. So the overall correlation was about 0.9, and then it was quantitatively more or less the same for all these different proteins in the two libraries. Now, these two libraries were both kind of drawn from the same underlying distribution of all these small protein domains. We didn't try to make one library that would be like completely different types of domains compared to the other. So I don't know how this would hold up. Like, I'm sure that the context of which other proteins you're around kind of affects how you aggregate, but at least, you know, it's not affecting it so much that we could take these 1,000 proteins, put them into a different group of 9,000 other proteins, and see quantitatively the same level of aggregation.
What's the concentration of the...
This was all done at 10 mg per mil. 10 mg per mil total protein in the mixture. So every protein is, of course, at a much lower concentration than that because there's, you know, 10,000 proteins in the mixture. Please.
Did you quantify at all the range of abundances for individual proteins?
We can't do that because the MS is not, you can't accurately quantify one protein versus another in the same sample. Please.
Not that it matters, but was it just an isotonic simple salt buffer, or
Yeah.
how did you?
Yeah. I know. I don't know. It was probably like PBS or something like that. There weren't like glycerol or special stabilizers. Okay. So next question. Does aggregation in these mixtures correlate with aggregation of individual purified proteins? And so I'm sure, well, I don't know if people have looked at this. Like when people do like cellular proteome, like thermal proteome profiling stuff, they don't usually then go like purify some of those proteins and see what's the difference between the cell lysate and the individual protein. But we did. So we purified, I don't know, like 15 different proteins. And so this is the amount of aggregation of all these different proteins at 50 C in the high throughput experiment with 10,000 other proteins in the mixture. And this is how much they aggregate just by taking like a soluble concentration measurement. When we purify a protein, we're not doing proteomics anymore. We're just heating it up to 50, cooling it back down, removing the precipitate, and measuring how much protein is still soluble compared to what we started with. And so this is 10 mg per mil of the whole library. This is 10 mg per mil of the individual protein. And we see like a pretty good correlation between how much these proteins aggregate by themselves and how much they aggregate when they're mixed in with 10,000 other proteins. We saw the same thing at 75. So it's not a perfect linear correlation, maybe because we might be able to measure a wider dynamic range. We might be able to measure a wider range on the individual measurements compared to what we could measure in the pooled. Or maybe there's something about having these proteins by themselves that actually causes them to aggregate more than when they're mixed in with all the other stuff in the mixture. But still, the rank correlation is pretty good, and the linear correlation is good. It's just not quite y equals x. Oh no, the rank correlation is better than the linear correlation. And we also saw the same thing at low pH, although once again at low pH, we observed a wider dynamic range on the individual measurements than what we did in the high throughput measurements. And you know, all these individual measurements were done like in triplicate, and they were pretty reproducible. Okay, so we can learn something about how proteins aggregate by themselves, even when we measure them in a weird mixture of a bunch of other protein domains. Okay, so we now have these quantitative aggregation phenotypes for all these proteins. Again, what can we learn from that? So one of the things is that the aggregation that we observed it correlated somewhat, but not incredibly strongly with simple properties of these proteins. So this is aggregation at 75, and this is a hydrophobicity scale. There's lots of hydrophobicity scales. This was actually the one that correlated the most with our data. So the higher hydrophobicity proteins were more aggregation prone than the low hydrophobicity proteins, but there's really high hydrophobicity proteins that basically don't aggregate at all in our experiment, and some lower hydrophobicity proteins that do aggregate quite a lot. So it's not like a trivial linear thing of this property. And the pH data was actually even more surprising. So this is aggregation at pH 4 versus the PI of the protein. And so of course, a lot of these acidic proteins are showing high levels of aggregation because they become neutralized at low pH and then they're more likely to self-associate. But like some of the basic proteins are aggregating at the acidic conditions. And even some, of course, there's lots of very acidic proteins that are not aggregating. So there's a lot of variation in these data that can't be tracked just by simple properties. So we tried building some machine learning models. One of the first machine learning models we tried was just predicting aggregation based on amino acid composition. So add up the amounts of all the different amino acids. And for we built linear ridge regression models and XGBoost, which is a nonlinear model. And for 75C aggregation, we could explain maybe like a third of the variation in the data just from amino acid composition, or maybe a little bit, about a third also for pH 4 aggregation. If we compute a bunch of features of these proteins, like hydrophobic surface area and hydrogen bonding, or like the radius of the AlphaFold model, or all the features that we could think of to calculate, you get a little bit better at predicting the 75C aggregation. You can explain 40% of the variance. The acidic model also got a little bit better. But actually, the best model was, once again, actually fine-tuning the assay protein language model was able to explain about half the variance in both of these data sets, more than half of the 75C aggregation and a little bit less than half of the aggregation that we observed at pH 4. One thing that I really want to do is to take our folding stability model that Yalen made and then fine-tune it again on these aggregation data and see if we do even better with a model that really understands folding stability deeply, but then is also trained to understand aggregation. Okay, but anyway, without doing that, we can explain about half the variance with this fine-tuned saprot model. So this is what the model actually looks like. It shows good correlation with the 75C and the pH 4. It can't predict like the absolute strongest aggregators. So the difference between -3 and -5 is kind of lost on the model. And of course, there's some noise around these because they're only R-squareds of 0.5. Okay, so this preprint will hopefully be shared in the next few weeks. This manuscript's really ready to go. I'm excited to put it out there. And it's really nice to see that we can learn about how these proteins aggregate by themselves, at least under these particular stresses, from a pooled assay on 10,000 different things. And we have lots of directions where we want to go with this. So we want to scale up the assay. 10,000 was just a nice number to try, but we actually just bought a library of 100,000 things, and we're going to see if the mass spec can quantify all these 100,000 different proteins in the mixture, or like how much can we scale this before we can no longer quantify these proteins by mass spec? We want to figure that out. We did this all on these small domains because I think they're really easy to analyze, but of course we want to apply this to nanobody domains or more diverse, larger proteins. We want to expand the stresses that we've looked at. These were just the first things and the easiest things to do, but if all these other stresses on proteins are really important, and they're all going to probably teach us different things than just what we learned from the temperature and pH stress. We didn't actually try to use our model to do any protein engineering yet, but what happens when we take the assay prop model and kind of prospectively test it on designing aggregation resistant variants? I should also say that if we use subsets of our data, then, and like what happens if we train the assay prop model only on 25% of our data or 50% of our data? We have not yet saturated the ability of this model to predict what we're measuring. So using like all of our data leads to a substantially better model than using 75% of our data. So we know that there's easy improvement just from collecting more data, which we want to do. Yeah, I'm sure all these proteins are aggregating through different mechanisms. Some are probably aggregating from the folded state, some are unfolding, some are aggregating like during the process of refolding or forming amyloids. There's all this complexity that I feel like we should be able to tease out. And so I'm really excited to keep doing these types of experiments. Okay, I'm basically done. I just want to close. Well, yeah, why don't we take questions and I want to close up on a few other ideas.
So I mean, the obvious question for me is soluble aggregates. Do you have a
Yes. Yeah, I think we should just filter out soluble aggregates either by SEC or something else. We will lose more of the yield that way, and we'll have to have larger amounts of protein going in. But yes, it's totally a smart thing to do and important.
By the common features like hydrophobicity scales. I guess interesting, but maybe not surprising. So one of the most common tools we use is like SAP or just
Yes.
hydrophobic patches.
Yes.
So how well did either of those do for explaining?
We analyzed like 10 different predictors on these data, and I don't have a slide on it. It's in the paper. I'll pull it up at the end and show you what it looks like. None of those tools were better than maybe like 0.3 or 0.2 on these data.
So
a little better than random. Actually, you know what? Let me show my last slides and then I'll take all your questions. I'm giving a talk at MIT Bioengineering tomorrow, which is actually going to be on a totally different topic. We have a really cool preprint out where we actually measured conformational dynamics of proteins through hydrogen deuterium exchange experiments, if you know what those are, in a high throughput way. So we actually measured the energies of conformational fluctuations for 5,000 different domains. And this plot maybe doesn't make any sense to you right now, but if you come to my talk tomorrow, then it'll all make sense, hopefully, by the end of that talk. Or you could look at this preprint, which was a project that was really long in the making to be able to measure dynamics for many different domains. And I'm really excited about this work. So I don't think there'll be free pizza though. So like, you came to the better one. Anyway, please come tomorrow here at MIT. I also really want to thank OpenFold and all the sponsors of OpenFold. And one of the reasons why OpenFold is so important to us right now, I don't know why that was highlighted. The NIH currently owes Northwestern like $300 million, which they are refusing to pay. So, you know, all of you hopefully know about how like all these grants at Harvard were canceled and then reinstated, at least to some degree. Northwestern's grants were never canceled. The federal government just stopped reimbursing our spending on our grants. So we have active grants where we spend money, but then they don't reimburse us, which is the way that all of NIH normally works. Like when you get a grant, they don't just put a bunch of money in your bank account. But so Northwestern is in big trouble right now, and that's causing a lot of restrictions on how we can spend money and hire people. And so if anyone wants to collaborate with my lab, I'd be really excited to look into those projects. But also one of the things that's really helping us keep going is OpenFold is supporting a lot of this work, especially our work on expanding the folding stability measurements so that we can measure folding stability for larger domains and a wider dynamic range and truly get to the point where we have a model that can predict folding stability for any arbitrary protein sequence. So thank you, OpenFold. Thank you to all the members of OpenFold. If you're not already a member of the consortium, you should join. And yeah, the NIH sucks right now. And then last, of course, I want to thank all the people in my lab. Sydney, who did all the aggregation work, Kotaro, who did the experimental proteolysis work, June and Ted, who are expanding the proteolysis work to larger proteins, Galen, who built the model to predict folding stability, and support from OpenFold and some support from the NIH and other collaborators, and the Pharma Foundation that supported Sydney's aggregation work. Thank you so much. I'd love to take your questions. He liked it, or she, he? She, all right. Please.
You mentioned getting like small protein things as models for so what we have learned or what hints do we have about how folding works in larger proteins? or just a bit different? Or is it a different problem?
I think that the principles of folding stability in larger domains are basically the same as the principles of folding stability in small proteins. The biggest challenge for our model right now is not actually the size of the protein. It's that a lot of the model proteins that have been used for folding stability studies in the literature, like T4 lysozyme or staph nuclease, are incredibly stable proteins. They have folding stabilities of like 10 or 15 kcal per mole and stuff. And when we have a model that's only ever seen proteins in the like 0 to 5 range, it's hard to predict that this other protein has a folding stability of 15 kcal per mole. So I think one of the reasons why our existing model struggles on large proteins is not because the principles are different, but because it's never seen stabilities in that dynamic range before. I don't know if that really answers your question. We could talk about it more after. Actually, I also want to just, since Vanita already asked, let me just pull up this plot. So here is the answer to the question that was already asked about the correlation of all these different things. No, it's not. I'm sharing it on Zoom. Maybe if I move this, there we go. This is how much all these different predictors correlated with our aggregation measurements. So SAP, that's this. SAP was actually the best predictor of the pH4 aggregation, and CAMSOL's pH4 was the second best. SAP was actually not very predictive of the 75C aggregation. I don't know, go figure, please. Of the aggregation?
Yeah.
It's very high. I don't know what the number is, like 0.9 or higher, or something like that. Please.
Yeah, like absolute stability model also predict the substitution energy.
Yeah, it predicts the effects of substitutions about as well as thermal MPNN. And it can have problems with larger proteins because if the wild type is predicted to be like the max of the range and the mutant is the max of the range, then you're predicting a DDG of like nothing, but that's a weird artifact of the way the dynamic range works. So at least so far, we didn't see an improvement at DDG prediction from doing this. But I actually think that once we have this more comprehensive open fold data set that we're trying to make right now, including larger proteins, I hope to get better at DDG prediction. Please.
What are the technical barriers to measuring that?
For folding stability, the biggest barrier, well, the two biggest barriers for folding stability for larger proteins were the fact that the range gets a lot bigger, the range of stabilities gets bigger, but I actually think that we're in the process of solving that now, thanks to the new like denaturant-based approach that we've developed. The other barrier to doing it at scale is actually just the cost of DNA synthesis, of making all these diverse protein sequences for things that are either require really long oligos or are longer than you could fit on a single oligo. But I also think that there will be ways to get around that. So I don't think it's insurmountable, and we're working on all of it right now. Please.
Is this a software model using like structural tokens, or is it just basing it off of single?
It's using structural tokens, and we actually analyzed for the aggregation data. So we use like AlphaFold models to generate the structural tokens. And for the aggregation data, we trained it with and without the structural tokens, and it performed better with the structural tokens. Please.
Curious about the chemical denaturation. Did it not interfere with your proteolysis, or did that?
We use thermolysin when we do that, and it does start to lose some activity, but I think we're still measuring things well, at least up to 8 molar urea, and if we add enough thermolysin, then even up to like 6 molar or 8 molar guanidine. Please.
When you have
larger, the more complex library now, you might want a second degree of separation. Then you consider instead of just 2 points like, high temperature and normal temperature, with like a column chromatography, like a hydrophobic column.
Oh, I have like that totally makes sense. Basically to have a huge library, but then separate it out and do a bunch of different mass spec runs of different fractions of that light. That's what you're saying, right? Yeah, different fractions. Yeah, that would totally make sense. We hadn't thought of that. We're doing proteomics without a license.
Gabe, I'm wondering if you clustered your 20,000 proteins by structure, by TMLI, if you would have better correlation within a structure, or within a cluster, I mean, for these predictions. Like the same with the delta G.
So in the I don't know. I mean, the delta G predictions are already pretty good across clusters. So we didn't specifically analyze how it is within clusters, but it's worth doing. It's a good idea. In the aggregation, we did actually start to look within clusters. I don't know if we There's an entire figure on this actually, but I don't. Okay, so what does this say? Oh no, this just says the fold change. This is the correlation of some sequences in one particular cluster. I don't know. I don't think we tried looking at what are the correlations within the clusters aren't very large in this case. Maybe like this is one of the larger clusters, I think, and there's like 15 points in this cluster. But that's actually a good idea to see what's the distribution of correlation within these clusters of related folds.
Yeah, these are clusters by sequence.
Clustered by sequence, yeah. Please.
So it is aggregation. Do you see evidence of pickles and like PhD?
So everything's mixed together. We get one pellet, and we have not tried doing anything to that pellet. Oh, no, we didn't try, but that's an interesting idea. Please.
And I wonder your thoughts on how much the in vitro measurement explains what happened in vivo, because I think like when cells are producing protein, actually also like full translational folding and like how would you, is there any idea like can we measure things?
Yeah. There's probably some ways that they're similar and some ways that they're not. We haven't done a lot. We haven't tried to compare to anything that people have studied in vivo. The closest thing to that, which doesn't really have anything to do with protein synthesis, is just looking at existing databases like there's this Meltome atlas where people did similar experiments on cellular proteomes, and they also trained an aggregation predictor using saprot based on that Meltome data. And that was not a great predictor of what we observed in vitro. Going the other way, we didn't try to. Going the other way, I don't know that our stuff predicted what was observed in Meltome all that well either. I think we did that experiment though. I don't remember what the result was. Please.
You mentioned some of the singleton experiments were measuring more fold change than the.
Yeah.
I'm wondering if there's any limitations on mass spec testing for a library size.
Yeah, that's a good question. We only included protein in this analysis where we were actually able to quantify that protein in all the mass spec conditions. So if we basically don't detect the protein at all, then I think it gets lost in our analysis. I'm not enough of an expert to say how that played out. But you're right that it's something we should think about. It also kind of makes sense to me that if you have a really aggregation prone protein, you're going to see more aggregation when it's by itself compared to when it's mixed into this matrix of other stuff that doesn't aggregate as much as itself does. But yes, I need to go back and make sure I understand how the MS goes. I'll keep taking questions as long, or we could let people go and people can talk to me.
I'm just curious, since these are kind of like random pulled out.
Yes.
I don't know how much analysis you've done. Are there any heat shock proteins or other like chaperones that are in these mixtures?
There may be domains of chaperones, but they're only they're not larger than 80 amino acids. What really does exist here actually is almost by accident, about like 5% of these domains, or maybe even 10% in these libraries, are actually predicted to be unfolded, or to have pretty wide, or to have some substantial unfolded segment. And those kind of IDP or IDR like things could act as chaperones toward the other proteins in the mixture, but it's not. I mean, most of the stuff are folded domains in the libraries. Okay. Thank you for coming. I will happy to take people's questions as well.